Okay, so we'll begin a very exciting session about modeling plant responses to climate changes or to climate in general. And Amnon Erez, who actually, as far as I'm concerned, is the founder. I know there were people before him, but we started working on his models when we learned about chill, either in the academia or later when I did actual research. So he will talk first, and then Eik Ludwig will speak. He did a lot of work about adapting these models, this notion to in this case to California first, to multiple varieties, to test different models, to test Amnon's model and compare it to other approaches. And then Mache organized a joint talk of me and him, where we strongly encourage you to participate while the talk is going. So basically, we wouldn't want to run through our talk without any comments and interpretations. So Amnon, are you ready?
Yes, I am.
One more thing. Amnon, one more thing. People can write questions in the chat, so we will collect this question and maybe we'll summarize them at the end because we don't want
to it's best after the talk, yes.
Okay.
Okay. So I'll share. Okay, so good evening to everyone, or maybe in other places it's a good day. I'm going to talk today as an introductory presentation on dormancy, chilling requirement, and the dynamic model development and potential improvements. I first would like to thank the organizer for inviting me to give this talk. Just a minute. Yes. First, I'll speak about the climatic adaptation of temperate fruit crops. These are the crops we worked with, and how they are adapted to the specific climate where they grow. These fruit trees were selectively adapted to grow in areas where sharp seasonal climate exists, and the winters are cold to very cold. Trees were adapted to grow in spring and summer, stop growing in autumn, and prepare for survival in the coming winter. The summers are typically with long days and mild to warm temperature. The day length changes markedly in spring, increasing, and in autumn, decreasing. Climatic adaptation of temperate fruit crops. The critical element for these plants were, 1, how to know that winter is approaching and growth should stop. 2, how to prepare for survival during the cold period. And 3, how to know when winter is over and growth and development can be resumed. So how to know that winter is approaching and prepare for survival? 3 processes develop generally in a simultaneous way, enhanced by shortening days and cooling weather. They are stop of terminal growth, developing of dormancy in the buds, induction in the dormant organs of cold resistance, and in temperate zones, leaf drop occurs in parallel. How to survive cold winter? For that purpose, dormancy was developed. Basically, dormant organs develop the capacity to stand at very low temperatures without harming the dormant tissue. Specific tissue has to be developed for survival, not for growth. The definition of dormancy is that dormancy is the sum of processes that constitute a programmed inability for growth in various types of plant material, meristems, in spite of suitable environmental conditions. How plants know when winter is over? The dilemma. While dormant buds having a meristem can survive very low temperatures, Once bud starts to develop and grow, cold resistance is lost. This process cannot be reversed. So if premature bud break occurs, all buds may die. So nature selected a mechanism to overcome this problem by measuring the duration of winter. This mechanism is chilling requirement. Every species of the temperate fruit crops and every cultivar has a specific chilling requirement. Only after this chilling is reached will the buds respond to favorable conditions and break a bud and will grow. There are adaptations between the cultivars growing in a certain place and the chilling normally available there. Chilling is a specific combination of temperature, level, and duration, as I will describe. Effect of proper dormancy breaking. When dormancy is breaking properly, three aspects can be apparent. High level of bud breaking, uniform bud breaking, and early bud breaking. The temperature effect of dormancy. I'm going to describe the basic effects of temperature on dormancy in this presentation, but they can be divided into three: the low temperature effect, the high temperature antagonistic effect, and the moderate temperature promotive effect. This work was done in the US with cooperation with Dr. J. Cuvion in Georgia, using rooted pitch cuttings and controlled conditions. The advantage of this plant material was that we could produce thousands of plant material, identical plant material, and use it under controlled condition. And by that, we could get enough replicates to get reliable results. Here, for instance, you see the effect of high temperature in a daily cycle with a maximum temperature of 15, 18, 21, and 21. 24 continuously was a sort of control with no bud break. And here we have continuous chilling at 4. So you can get an impression that 6:15 may be even better than 4. 6 to 18 is about equal to continuous chilling, and 6 to 21, there is a full negation of chilling. By that, we got the following results. This is the low temperature effect. We can see the effect of the same duration of exposure in all this temperature. The duration of exposure was exactly the same, and we could see the level of bud breaking after moving the plants to forcing conditions. And you can see a very clear optimal curve for leaf buds as well as for flower buds. You can clearly see that at temperature of 14 and higher, there was no bud break, which we tested actually till 20. While towards 0, there's a sharp decline, but still at the 0 degrees, we can get some effect much lower than 6 or 8 degrees. Now, let's look at the effect of high temperature. There's a strong sensitivity of dormancy breaking of buds to small temperature increases. Here we see cyclic temperature of 16 hours during the day, 16 hours of chilling and 8 hours of warming condition. And we compare continuous chilling for lateral vegetative buds and for flowers. And cyclical temperature, as I said, 16 hours cold, 8 hours high, 4:15, 4:19, 4:20, 4:21. You can see that the difference, first of all, you can see an advantage of 4:15 over 4, at least for vegetative buds. Then you can see that there is a very sharp decline at 4:19, 4:20, and 4:21 is almost 0. So the sensitivity is very high to high temperature, and increase to even 20 degrees almost nullifies all the chilling given. So this is the definite effect of the high temperature. Okay, now here we see the effect of moderate temperature on breaking dormancy in flower and vegetative peach buds. Here we checked, we had also a diurnal cycle. As I told you before, here it was 6 degrees at the low temperature for 16 hours and higher temperatures for 8 hours. So we got this curve. Here is again, the amount of chilling was exactly the same in all cases. And you can see clearly that there is a curve that goes up from 4 degrees, continuous, that goes very high with vegetative buds, and with the dashed line is flower buds, dashed line with closed circles. But since the temperature of the temperature of 6, of 9, 11, 13, 15, not 15, not, but 13 has some small effect on dormancy breaking, we deducted that and we got these open circles, which is the actual effect of the moderate temperature. And you can see clearly there is an increase of effect of this moderate temperature between continuous 6, for instance, to 613, very high effect, almost double the effect. So it's not a negligible effect. It's a very clear effect that you can see both for vegetative buds and for flower buds. So we term this the effect of moderate temperatures, and we'll explain clearly, it will be very clearly understood why we get this effect. So if we'll summarize the chilling requirement based on work under control conditions with peaches, follow the 3 temperature effect that were defined, the favorable effect of chilling with maximal effect at about 6 and no effect at 14, negation of former chilling by temperature higher than 18, with negation effect stronger with duration of heating in the daily cycle and with the level of temperature. And we have a moderate temperature effect between 10 and 16 that shows synergistic effect with chilling temperatures. The dynamic model was developed on the basis of these results. It was developed by Dr. Fishman from the Volcani Center, and we have the model that we published on the name of us, Couvion, myself, and Fishman. Now, the dynamic model explains the various effects of temperature on dormancy, leading to dormancy breaking. It is based on two reactions. The first produces an intermediate, which is reversible. The second produces a fixed product. It is based on data that shows that once we have temperature, low temperature, over a period of 28 hours, we get a fixed effect. In a daily cycle, we never get a fixed effect. But if you have more than 1 day, 3 days or so, we do get the fixed effect. Now, the second product is a fixed product. The second phase is a fixed product. The fixed product is accumulated by fixed quanta. Every bud has its specific requirement of needed quanta. We call it chilling portions, needed for completion its chilling requirement and allowing bud break. Here we see it this is a demonstration of the effect. A reversible process with chilling temperature promotes the buildup of the intermediate, while high temperature reverses it. Once we get a certain critical level of the intermediate, we get a chilling portion produced, which is a irreversible fixed effect. And this is the model that was developed. And we can see here the intermediate X. We have a K0 and K1. K0 is advanced by lower temperature, K1 by high temperature. And in every temperature, we have a certain steady state level of X. And K produces once X reaches a critical level, we get an autonomous move of one portion that is fixed. So I won't go into details here. It was published many years ago. You can see it. And now I want to explain what does it mean. In every temperature, we have a steady state level of the intermediate. The higher the temperature, the lower the steady state level. And you can see here the steady state levels of the intermediate between 24, 20, 16, 14, and 12. 12 is the only temperature here where it reaches the critical level, and therefore there is an accumulation of quanta of portions, of fixed portions. When you have temperature lower than 12, the curve is even steeper, and the critical level is reached earlier than 12 degrees. And when the temperature is very low, then it takes more time for reaching the critical level. This is a simulation of chilling in portion at three temperatures, 6 degrees, 12 degrees, and 14 degrees, for a duration of 240 hours, 10 days. And you can see that at 6 degrees, there is continuous temperature. You get an accumulation of the intermediate till it reaches the critical level. One portion is accumulated, and it starts to accumulate again the intermediate. By this way, we get accumulation of the chilling portions. 12 degrees, it does the same effect, only that the effect is slower, and therefore we reach the critical level in after a longer time, resulting in less accumulation of chilling portions compared to 6. When we reach 14 degrees, we are very close to the critical level, but the steady state level is below the critical level, therefore no portion is accumulated. But clearly, if we are moving from a certain point from 14 to 6, immediately the curve changes, and very shortly we are getting the critical level. This is actually the effect of the intermediate temperature that always are acting with chilling. The third simulation is of a changing of the diurnal cycle of 16 hours at 6 and 8 hours at 24 degrees. So at 6 we get this level of the intermediate, but when we move to 24, the level of the intermediate goes down towards the steady state level of 24. Then we go back to 6, 24, 6, 24. We give a lot of chilling, but there's no accumulation because we never reach the critical level. We check now what is the possible effect of global warming on deciduous fruit trees with results to chilling requirement and chilling accumulation. So increasing in minimum temperature under warming condition will lead to reduced chilling portion accumulation. This is very clear. In addition, there could be an increase in maximal temperature that will result in a higher level of chilling negation, and the combined effect will result in smaller accumulation of chill portions and increased difficulty in getting good bud break. In addition, for stone fruit species, damage to normal development of flower bud is expected. And this I'm going to show a little later. Now, there are other different practices that may affect the chilling requirement that were not taken into consideration in building the model, and I suggest to take them into account. The first one is the vertical growth compared to more horizontal or inclined growth. Vertical growth results in stronger apical dominance, and this has a very strong effect, as I will show immediately. Also, excessive vegetative growth increases markedly the chilling requirement of the buds. Here I show the effect of inclination or of vertical compared to horizontal or inclined growth. You can see a situation under marginal condition where inclined growth or horizontal growth will break considerably, while vertical growth remained, all the buds won't break, only the terminals will break. So horizontal bending may have a strong effect on improving bud breaking. Here we see the same effect on apple. Here we see trees, golden delicious apple trees in pots that were exposed to the same temperature, but were under forcing condition, were put either vertically or horizontally. And you can see an enormous effect of the position. This, what you see here, is the vertical growth that reduces very markedly the bud break. And here we see the horizontal positioning that had this much, much stronger effect. The excessive vegetative growth has also an enormous effect, a very strong effect. Here we see a low chilling peach tree that grew more than 2 meters in one year, and the buds on the very strong vegetative growth had a very high chilling requirement, very high, not to compare to normal growth. So definitely the excessive growth will increase the chilling requirement of a variety. As a consequence, reduced vigor will facilitate bud breaking under marginal conditions. This is very, very critical. Now, I would like to speak about an element that is the outcome of what we saw, and this is the terminal bud syndrome. Different buds require different chilling. The terminal bud have the lowest chilling requirement, then the floral buds or mixed bud, depending on the species. And the vegetative buds, lateral buds, have the highest chilling requirement. Lack of chilling for lateral vegetative bud, but sufficient for terminal buds, will advance terminal bud break, and thus will set apical dominance that prevent lateral buds from developing. So although those buds could develop, but because of apical dominance that is very strongly promoted by marginal chilling, that is enough for the terminal but not for the vegetative buds, will prevent the lateral from breaking, as I showed you in the upright vertical growth. Now, another element that we found from work done many years ago was the following. I want to show you here a treatment, a test, a trial that we did with potted plants. These were Red Haven peaches that were exposed to 1,200 chilling hours that we know is enough to break their dormancy. Now we have a continuous treatment, which was 50 days at 6. 26 before they were under condition that was not affecting chilling. So 1,200 hours gave, let's look only at the laterals, which are important here, gave a 57.5% of bud breaking. Now let's look down there. We have intermittent daily cycle. We have two cycles like this. One was 66 days at 6 to 18. We know that 18 has no negative effect. Only for 740 hours, we got 42% bud breaking. I assume that if we'll give them 1,200, we get about the same as we got with continuous. We have 75 days at 5 to 21. We know that 21 is negating chilling. Here we had only 1,000 chilling accumulated, and look what we got, 6%, which means actual complete negation. But here we have 1,200, in the middle, we have intermittent long period. Here we had 6 degrees for 38 days, 20 degrees for 12, 6 for 7, 20 degrees for 11 days, and 6 degrees for 5 days. And look what we got. We got 81%, much better than continuous chilling. So 20 degrees, when it is for a long period, we know it is not negative. It won't negate because there is already fixed effect. But still, why did we get this to 81%? To my opinion, what happened is that high temperature towards the end of dormancy starts to play a role. So the parameters are changing. So this, therefore, I call it changing parameters with dormancy. Late exposure to high temperature may have a positive effect. And this we never actually tested quantitatively, and I suggest that this should be one of the items to test in order to improve the model. Now I would like to speak about the last element, which is very interesting and very important, that normally is neglected, and this is specific effect on floral buds. After all, we are growing these trees to get fruits, and for this we need the floral buds. Now, floral buds in stone fruit species are very sensitive to climatic condition. Extreme condition will damage the buds and may cause death. The floral bud completes its development only after complete chilling accumulation. So during the whole dormant period, it's not complete, and it developed only when chilling accumulation is completed. Specific climatic conditions, especially heat spells, even in winter, will interfere with normal flower bud development. Under such condition, completion of chilling requirement will not improve fruit set and yield. Because when there is a damage to the flower bud, no matter how much chilling you give it, it won't reverse the situation. I want to show just a few pictures here of a situation like that. Here we see Somerset peach. Over here you can see the normal fruit, but in many cases you get what we call mummies, undeveloped fruitlets that were still attached but have no value commercially. Here we see similar effect on nectarines. This is a normal fruit, and many of these fruits are also mummies, are fruits that were not developed properly because of damage in the flower bud because of the heat spell. Now I want to show you some pictures of cherries. Here you can see a cherries, the same effect. You can see a normal fruit just here and there, and many undeveloped fruits that has no viable seed in them. Here you can see it very clearly. So this is also because of exposure to high temperature. Sometimes you get a very early senescence of the flower petals in these conditions. And here you can see another cherry treatment or cherry observation. Double fruit, which is very common under these conditions in cherries and in peaches and so on. And even a situation where there is no style whatsoever. You see a flower, you think it's a normal flower, but you observe it closely, you see there's no style at all. So this is an effect of a heat spell during winter. So here are my suggestions for improving the dynamic model. One, to investigate the change in parameters with advancing dormancy, as I mentioned. The other is to evaluate separately vegetative and floral buds regarding their chilling requirement, because there is a change in the amount of chilling they require. To examine the effect of growth vigor on modulating the actual chilling requirement. To test the effect of branch orientation on quantitative chilling requirement. And last, to examine quantitatively the specific high temperature value that may harm flower buds in stone fruit species and add it as a separate warning. Thank you. If there are any questions, I'll be very glad to answer.
First, unmute everybody. Unmute all.
There's someone in the chat.
So,
Unmute themselves.
who wants to ask a question first?
So do you consider ever the role of stems?
Again?
Do you ever consider the role of stem in the bud breaking, whether stems are involved in sending information to the bud? Because yes, stems. The buds cannot grow if there is no stem.
You speak about the branch, the stem?
The branch, yes. The branch, when it's being dormant, it doesn't function. And the bud, when it develops, it needs a stem on a big tree, especially that, let's say, 50-meter tall tree, because we're not always talking about the orchards. And entire system has to be functional to allow bud to develop properly. So what is the role of stem in this situation?
Okay. Well, one of the very special characteristics of a dormant bud is that that bud is isolated in a way from the rest of the plant. You can chill a single bud and this bud will break and the other buds on the same stem will not. So we know that the plasmodesmata are also disconnected during the dormant period. So this means that the effect is isolated. That's the reason why people, and myself too, we were trying to look at the effect of chilling of a variety by taking branches, cut branches from trees and put them under different conditions. I think that if there is no effect of wound, then there is the data are reliable. So the question with regard to stem, I know that you worked a lot about carbohydrates and the effect on bud breaking. I think this is more at the stage of bud breaking or before bud breaking. Of course, you can get bud breaking but no growth if you are disconnecting it from the tree, just having a short stem or like this. So actually, especially I think in grapes, they were using a single bud node to look at the response. But I think that I don't like it because of the wound effect. But otherwise, I think it's okay.
I have a question.
Yes.
I remembered you said that the vegetative buds that they need more chill than the floral buds.
Right.
But then, you know, all these pictures of heat spells during winter and anyway, and we know that these vegetative buds actually, well, they broke, these trees lived. So whenever you show this deformation in fruits or just fewer fruits and the mummy fruits, there were a lot of flowers actually.
Yes. Well, the point is that you may get damage to the flower bud if you get a spell in midwinter, very high temperature for a certain duration that I cannot quantify, but I know for sure. And what will happen afterwards is that the flower that is going to develop will be abnormal. So this tree got, let's say after the heat spell, it got enough chilling. And the bud break was looking good, but the flowers were abnormal and will never develop into a normal fruit. So this is a specific effect of heat spells. And since we are talking about global warming, I think this is one aspect that should be observed if there is any such condition that will cause damage. Now, I want to stress that this happens only in stone fruit species. In peaches, in plums, in apricots, in cherries, not in apples and not in pears. In apples, we got sometimes flower buds that didn't break, and they passed the whole summer at very high temperature, and they broke the next year and produced a normal fruit, but never in stone fruit species.
Okay. Are there more questions?
I have one question.
Yes.
I would like to know the efficacy of the dynamic model that has been presented by Professor Arrange. And the second part of the question is that what will happen if the temperature timing is either shift earlier or it goes beyond some time.
I didn't get the question.
Can you repeat the question?
Can you repeat, please?
What will happen to the stem buds if the temperature shifts either shifts here or to the positive side or negative side? The chilling part. I am talking about chilling of the temperature.
You give chilling at no, I didn't get it. Sorry. Oh, you got the question.
I have given in the chat section also the question.
there is a you send a chat.
Yeah.
Okay, either a change in timing of temperature change. If there is a change in timing of temperature change. What do you mean a change in timing, in temperature change?
Chilling timing.
Asking whether
you mean, for instance, let's say, you mean, for instance, if there is a short, let's say there is a 20 hours a day of 6 degrees and then only 4 hours at 21, right?
Yeah.
So here we had a very clear results. The shorter the exposure to high temperature during a daily cycle, the smaller the negative effect. When we get only 2 hours of 20 degrees, we didn't get any negation at all. When we had 8 hours, we get full negation. And if we go to higher temperature, like 24 degrees, if we have even 14 hours of 6 and 10 hours of 24 degrees, we get still negation. So negation, the strength of negation depends both on the length of the high temperature period in a daily cycle, as well as on the level of the high temperature.
Okay.
That's a good question.
Okay,
yes.
thank you.
Thank you. So now, Eik, do you want to speak before we show the... Eik actually prepared his video in advance, and Sophie will load this, but maybe you can open with some words.
Sure. Kann ich mal jetzt sein? My son is just entering the room. Hello, everyone. It's a pleasure to be here. So I usually have trouble with timing, and so I decided this time I'm going to pre-record so I don't start rambling about stuff that doesn't actually fit into the schedule. And so you'll get to see a video, but and I don't really need to maybe introduce things too much. I put that all in the video, but we can have a discussion afterwards.
Okay. Thank you. So, Sophie, can you start the video?
It is a great pleasure to talk to you today about analyzing tree phenology in the changing climate. My name is Eike Lüdling, and I lead the Department of Horticultural Sciences at the University of Bonn in Germany. I want to be talking about tree phenology, specifically about the phenology of deciduous fruit and nut trees that go through an annual cycle that I think all of us are quite familiar with. I am interested in how climate change may affect these annual dynamics that we see in fruit trees. Specifically, I'm looking at the dormancy season because even though the tree doesn't seem to be doing very much, it actually is quite responsive to temperature. And it's a fascinating period to be looking at when it comes to climate change impacts. Here's what happens during the dormancy period. Dormant trees must experience certain temperatures to resume growth. They have a chill requirement during the endodormancy period, which is the first phase of dormancy, and then afterwards they have a heat requirement during ecodormancy, the second part of dormancy. Between those, there's possibly some sort of transition phase. If we don't meet these requirements, we're going to have a hard time producing fruit or nuts. And we would like to project phenology changes resulting from changes in exposure to chill and heat. But this is pretty difficult. We need information here to forecast future phenology for a system of interest. So here's the system of interest, and here are the potential impacts we may be seeing. We require information here now on system exposure, so the changes we're likely to see, and on system sensitivity. How is it going to respond? Doing this requires reliable scenarios of future temperature. And this is not so easy because the best practices in climate science tell us that we should not be using just one scenario or two scenarios. We should be using a whole ensemble of scenarios, meaning outputs of different climate models for different representative concentration pathways, and we should probably be looking at different points in time as well. Pretty complex task. And we also need reliable models of the system's climate response, and that is also quite challenging in this particular case. And together, this actually ends up being a substantial challenge. It's a complex and rarely achieved in phenology modeling. There are very few convincing studies out there. I've also struggled with this quite a bit, but I think eventually I saw some land in this quest, and I put together a framework, and I decided I'm going to put this together as a package for others to use and make climate change easier for me in the future, but also for other people. This is the origin of the Chill R package for R, which assembles a set of functions that help us project agroclimatic metrics for future scenarios. Here you see an output that Chill R can generate for us. What is shown here is a climate change projection for winter chill for pistachios in Tunisia. What you see is chill accumulation and chill portions here on the y-axis, and in the first panel on the x-axis we have time. Dataset started in 1973 and it goes until I think 2018 or so. Every blue dot you see in this diagram is the chill of one particular year. Now there's a lot of interannual variation here. It always goes up and down quite a bit, and so I've added a feature here that allows us to produce summaries of the climatic situation at a particular time. So ChillR includes a weather generator, or it acts as a weather generator that somebody else developed, and it allows us to simulate 100 representative versions or more, as many as we like, of a particular year that we're interested in. Here, for example, we have the year 1975. This box plot here indicates the plausible distribution of chill that a grower at this time could reasonably expect. We see that most of this was above this green line here, which is the approximate chill need of pistachio. So about 90% of the years, a grower at that time could expect enough chill. But you see that this distribution has actually gone down quite a bit. And by now, if we don't have a 90% chance anymore, we're probably at something like 50% or even lower. So currently, in every second year, we should expect our pistachios not to get enough chill. And this has actually already manifested in some years where pistachios had showed scattered bloom. They showed no flowers at all in one year even. So we are seeing these effects actually in practice already. The other panels here show future scenarios. And in each case, we see outputs here of 18 climate models. And for each of them, we have such a distribution here, which is the same type as we've seen in the other panel. We have a moderate warming scenario here for 2 points in time in the future and a strong warming scenario. And in this case, it doesn't even really matter. We don't even have to look at in detail all these scenarios. We can tell that in pretty much all cases, we will not be able to produce pistachios anymore, assuming that our understanding of the temperature requirements during dormancy is correct. So these are quite alarming prospects for Tunisia, for pistachio growers at least. And we've done this similar analysis now for many locations, for about 20 locations or so in Tunisia, and for each of these we've made a plot like the one you've just seen, and we then interpolated this information to make maps of the chill distribution in Tunisia for several scenarios in the past that I'm not showing here, but also for a number of scenarios in the future. The prospects are quite concerning. We're expecting quite severe chill decline in this area throughout the century, and it seems quite likely to me that growers will have to make substantial efforts to adapt to these changes. So we have a fairly good system now for producing these temperature scenarios we need for future projections, but what about the models? I've already shown you this. This is roughly how the trees respond during dormancy. These are the things we have to consider, but the current models of this process are far from convincing. And we have chill models, we have heat models, we have a rough idea of how chill and heat possibly interact, but there's room for improvement in all of them. I very much appreciate the dynamic model for chill accumulation, but this was put together more than 30 years ago, and we've learned a lot in the meantime. And it also has, it's performed better than other models in pretty much all analyses that I've done, and that's quite remarkable, I think. But it still leaves a lot of data points unexplained, and a lot of observations that we've made aren't fully in line with the predictions of this model. So there's room for improvement. But how can we find information about the temperature response of trees? It is difficult because we don't really see any obvious changes going on in tree buds throughout the dormancy period. but we know that there's this endodormancy and the ecodormancy. So how can we even tell those apart? I've worked a bit on trying to delineate temperature response phases based on long-term phenology and temperature data sets that fortunately exist in a number of places. And the method that I've applied to this task is partially squares regression, where basically I'm relating the bloom dates or the leaf emergence dates or whatever they are of temperate trees to daily temperatures throughout the entire dormancy season. And the result looks something like this. This is an analysis of a data set on walnut leaf emergence in California. And what you see here is the coefficients of a PLS prediction model of bloom dates. The input variables here are the daily mean temperatures throughout the entire dormancy period. And while there's a lot of up and down here, which we should expect because PLS is a data mining technique and it does better with a lot more data than we usually have, so we'd expect quite a bit of noise here that should always be visible. But we do see two quite consistent phases here. One with negative model coefficients and one with positive model coefficients, and these can be interpreted as periods where high temperature delays bud break. So the higher the temperatures are, the later bud break occurs, and this is what I interpret to be the chilling period. And what follows is a contrasting period where warm temperatures actually have an advancing effect on bloom dates, and this is quite likely the ecodormancy period, the forcing period. So we can now identify the transition point or transition period between these phases, which allows us to now summarize the temperature needs in these particular periods. What we can also do is we can look at the phenology response to temperature during these chilling and forcing periods, and that's what's summarized here. What you see here is on the x-axis the temperature during the chilling period, and on the y-axis the temperature during forcing. And the dots here and the colors and the lines, that's the bloom dates, or the leaf emergence dates in this case. And we see the earliest dates here shown in blue in the upper left corner, and this is the combination of a cold chilling period and a warm forcing period. And the latest dates we see occur when the chilling phase is warm and the forcing phase is cold. What I want you to note here is these contour lines that are drawn that describe the shape of this response in this particular case, because we'll be seeing these in the next few diagrams that are coming up. We've also done such an analysis for apricots in China across a climate gradient. This gradient was comprised of these 5 locations here with quite strong differences in climate. We've done the same analysis here and drawn these contour lines as we've just seen for the California data set. In the coldest locations, especially where the chilling period is really cold, we're seeing these contour lines being pretty much horizontal, whereas the warmer the climate gets, the warmer the chilling period gets in particular, the more do these lines start sloping upwards. So now here in Guiyang, in the warmest location, we already have a slope of about 45 degrees. This led us to come up with a general hypothesis of the tree response to warming. And according to this hypothesis, the response depends very strongly on the temperatures during chilling. As long as the chilling phase is pretty cold, as is currently the case in Europe, for example, and in many other places, we would expect bloom dates, leaf emergence dates, to depend almost entirely on temperatures during forcing. Chilling period doesn't have a big influence. The warmer the forcing period is, the earlier do spring phases occur. But as the chilling period gets warmer, we see a delaying effect of temperatures during chilling coming in. So the warmer the chilling period gets, the later bloom dates are to a certain extent, but for quite a while this small effect is still swamped by the effect of warming during the forcing period. So we're still probably seeing this advance in bloom dates as a response to warming, mainly driven by the forcing response. But if the chilling period gets even warmer, at some point we're starting to see a situation where it becomes a dominant driver of bloom dates, and the timing of spring phenology depends almost exclusively on temperatures during chilling and almost not at all on temperatures during forcing. And the warmer it gets, the later the spring phases actually occur. Not earlier, but later. Now this is a hypothesis. We should now try and see if we find evidence for that actually happening. And to find this evidence, we're going back to Tunisia and looking at our pistachios again. We also have long-term bloom data from central Tunisia, so we could do a similar analysis. And this is among the warmest locations where pistachios are grown. And these are the contour lines, and they are indeed pretty much vertical. So we're seeing basically what we predicted with our general theory derived for apricots in China. These are different pistachio cultivars, and for almost all of them, we're seeing these lines being vertical or pretty close to vertical. So this is actually happening, and the concerning question is what happens when we move to even warmer chilling periods. And I've already said this earlier, we're actually already seeing what happens then. The pistachios start showing severe confusion, they start showing scattered bloom, they start showing all the signs of lack of chilling, and eventually, in extreme cases, they may no longer flower at all. Now we have some idea of the endodormancy and the ecodormancy phases, when they are, and the temperature response, but we still don't really have a phenology model. So how can we make better models? I think we have to still make improvements on existing modeling frameworks. So far, I think we've not fully exploited what we already know. For example, the dynamic model is out there. It's been used in its pretty much original form. In most cases, we're not able to parameterize it properly. There's a lot of work that still needs to be done in order to fully exploit the potential of the things that are already out there. So we're currently putting together such a framework, so hopefully we'll have something for you soon on this front. But we also, and maybe that's a bigger challenge, we have to synthesize our understanding of the biological processes during dormancy in order to make process-based models. Because as great as the dynamic model is, it was developed more than 30 years ago. And imagine the progress in biological understanding that has occurred during that period. We currently have no models that actually make use of this wealth of knowledge in a way that allows us to make predictions of bloom dates or leaf out dates for our trees. We've started putting something together. This is work by Erika Fadon, mainly, who works in my group, and she started assembling the different processes that we should consider in making a process-based model. The actual model is still pretty far away, but we're starting to make some baby steps towards that goal. What we also have to get much better in phenology modeling and phenology predictions is to express our uncertainties. There are lots of studies out there that come across as if they were able to precisely predict the bloom dates of a particular tree in a particular place in 2080. We cannot do this. There's a substantial error because there's a lot we don't know about dormancy, and our models certainly are very far from perfect. And we have to get much better at expressing this uncertainty because farmers need to know. Farmers need to know how certain our predictions are. They need to know what the risks are. And we kind of owe them information on this and honesty in translating our uncertainties into error bars that accompany our projections. Some take-home messages. State-of-the-art projection of climate change impacts on phenology is quite challenging, but the Chill R package makes this job easier. So if you haven't used it yet, please feel free to check it out and see if it's useful to you. We also need better impact models that are informed by observations, and we need models that are based on actual biological processes and what we know about tree biology, rather than on the abstract concepts of chilling and forcing that we're still, even after decades of research on this topic, having a hard time of mapping to actual biology of the tree. This is what I wanted to tell you today, and I thank you for your attention. I believe we can have some questions now. All right, and I still believe that. So, do you have any questions?
So who wants to go first?
I would like to ask a question. Okay.
Yes.
Well, the question is, Eike told us about the effect on bloom time as a parameter, whether it is affected by forcing conditions or by lack of chilling or by chilling. But I would challenge this, and I think that in order to know first if chilling was enough to break dormancy, like in pistachio in Tunisia, the best way is actually to know what is the actual chilling requirement and to follow that, and to look more at the level of bud breaking on the tree, like percent of bud break, time of bud breaking, uniform of bud breaking as a parameter rather than to look at the time of bloom. By this, you can get, to my opinion, a better quantitative value.
Mm-hmm. I think that's certainly true, and I mean having more detailed information on what exactly happened in trees would certainly be nice. I mean, what we've also experimented a bit with is to kind of model a tree as a population of buds that break at different times, and to basically try and predict the population dynamics of this population by looking at the chilling and forcing accumulation. I think there's a lot of potential in that. It's a little bit more complicated. We sort of stalled a bit in writing that paper at some point, but I think there is still a lot to be explored at that level. I mean, the problem is that most data sets kind of give us information on the whole tree only, and maybe only the full bloom dates. And we know there's always an error because it's kind of somebody's personal judgment, and there's a lot of imprecision there. And more detail would certainly be nice, but of course that would take up more effort to collect.
Yeah, I think that the effect of terminal apical dominance can come, this is a very important element when you start to get into not enough chilling. Like if you look at the situation in Tunisia, I'm sure that it will exacerbate the situation, because even if you had less chilling, that normally if you would decapitate the branches, you are going to get normal bud development. But if you leave it and the terminal bud starts earlier, it will prevent the other from breaking. And the effect is going to be much worse than otherwise.
Interesting, yeah.
So,
worth a try.
Okay, so your diagram of chill versus heat as a limiting factor.
Mm-hmm.
So, is it that we speak so much about chill because that's mostly the case where we grow these specific crops we're now interested in? The chill is the limiting factor because these places are not very cold, and or is it because we just don't know enough about the second phase, or is it because we don't know enough? But like we're always speaking of chilling, and now question is, should we speak so much about chill or?
I guess to some extent heat happens automatically at some point. It always gets warm in spring, so it's not such a limiting factor. And I mean, of course, I mean here in Jerusalem, we're more concerned about the chill part. In Israel, in Tunisia, and those places, California, Spain also. I mean, here in Germany, we're not particularly concerned about it, and it doesn't seem like it's going to be a limiting factor anytime soon. It is certainly the more interesting part to be looking at. I also think, though, that we probably should at some point also look at our heat models. To what extent that is credible, I'm not so sure. I mean, I think there have been some experiments that this was derived from, but it's also not that much. Like sometimes when you look at the history of these models, you don't actually, when you get to the end of the citation line, for so to say, you don't really find very much. I mean, that we have, so I'm really glad we have the dynamic model, because when you go to the end of the line of the citations of chilling hours, there's really nothing at the end. So at some point somebody claims to have a model, or not even, somebody just mentioned that this may be happening, and then it gets cited over again, and it becomes biological truth at some point. And I suspect that if we look at the heat models in detail, we may also have not find much. So what I actually do suspect is that we're using this, I mean, it seems like we're using the same model for dormant buds that we're using for growing trees during the growing season. And to what extent is that credible? Yeah, when the tree has leaves and stuff, I'm sure the growth dynamics will be driven by photosynthesis and by certain processes that just aren't happening in dormant buds to that extent. So I think at some point we may have to revisit that as well. But it is pretty difficult because these buds don't change. Don't change much. Yeah? So they're just looking the same way throughout this whole period. So how do we properly model this? And it really requires the kind of systematic experiments that Amnon has done in the 70s and 80s to do this. But who's going to fund these for us now? I don't know who did it for you then, but where do we find the resources to do this kind of stuff?
Yes.
Okay, we have to continue. Or would you like to present the next one?
So the next to speak is Maciej.
I thought it was or you starting, yes or no?
No, I thought we can skip my intro since we so Maciej would speak, but that's a joint effort. Me and Maciej came upon studying chill and then later sugars because there was well, pistachio trees were dying in California Central Valley for a while, and they were actually dying in warmer winters, but they were dying of frosts. So we started looking into chill acclimation, energy balances, and at some point we realized that temperature kinetics drive a lot of what happens within these trees during winter. We never suspected this would go on into modeling phenology and bloom times and climate effects, but it did. So I think Maciej has a very interactive presentation to you.
Okay, so yeah, as Or said at the beginning, the effort was an interesting element that required a lot of coffee. This is coffee and a lot of sugar. And we just, I will start my presentation here. And what is important right now for this presentation is to interrupt me whenever you don't agree. And also what I would like you to do is, I really hate talking to a cemetery, okay? That looks like a wall of dead people. I'm sorry. If you can turn on your cameras and you're not ashamed, please do it, because it's much nicer to talk to faces than to just names, okay? Okay, I start broadcasting to my iPad. And okay, there we go. Where is it? Okay, so what I call this talk, what we call the talk with R, we call it winding up the bloom clock. The sugar levels at senescence determine how trees respond to climate shifts, or whether they bloom or not. And the goal of this talk, here it is, goal of the talk is to present and discuss possibility that carbohydrate metabolism during tree dormancy is a part of internal clock mechanism that does the decision making, okay? And so just for the rest of the talk, I will use this SC symbol here as my, as a soluble sugars, and sometimes I use ST as starch.
It's soluble carbohydrates.
Soluble carbohydrates, yes.
Because Maciej would not allow us to use SS as an abbreviation ever in the
yes,
work.
that is true. I did not allow to do SS.
I was in. But...
Okay, here. So what is the idea that started us? So we talk with Or a lot about carbohydrates, and this is like the theme right now of my lab. And one of the interesting point of carbohydrate or soluble carbohydrates in the cell is that we believe that cells maintain certain level of carbohydrates inside the cells. And doesn't matter whether it's summer or winter, they have, for summer or for winter, they have certain level that they have to maintain. It's a similar way as we're doing it in the, in humans, we maintain certain level of glucose. And this level goes like up and down, and whenever it approaches the limit, there is some sort of a cellular activity that will change the direction of soluble sugar concentration in the cell to, let's say, decrease it. And then it goes to minimum, it will increase it, and so on. So in the cell, what happens in the cell? In the cell, we have this soluble sugar, soluble carbohydrate concentration, and this concentration is, variable due to the environment. But where it goes? First of all, there are losses. So there are losses, and this is like respiration. Then we have building blocks for cell maintenance. And if so, these are like two major things: respiration, and then there is also potential for export. And if so, these are like two major things: respiration, and then there is also potential for export. Why there is a talk? So these are like two major. Why am I talking when I don't know why there is a background. So there is.
You wanted somebody to interact with you.
Oh yeah, but it was myself.
Yeah.
Okay, and there is also starch in the cell, and so when there is too much, so losses is also when there is a synthesis, starch synthesis. On the other way, what are the gains? The gains is the import and the degradation of starch to form sugar. So this is basically the dynamic of the cell. And then during the winter time, most of this part, like most of those cells became relatively dormant. It doesn't matter whether they're in the bud or in the branch. Our focus with ore is to see the tree not as a bud, as and a branch and a root. It's not. We see it as a system approach. We see it as a whole organism that has to decide what to do together. It's unlike some people believe that there is this bud, they make a decision, they separate it, they do this, but they cannot grow without the branch. Branch cannot survive in the future without the bud and without the root. So all this whole system, the entire tree is like one big functional system. And that's how we see it. So there is this interactive part of it. So all those cells, they have to gain our information. The local information is very, very similar for everybody. It's not like bud sees the world differently than the branch, and branch sees the world differently than roots, and so on. They have different environment that they live in, but they see the world very similarly. And they have the same genetic mechanism and so on. So in the winter, you can say that respiration is maintained. So building blocks, export and import, especially import in the winter, those things are not, okay, we can do the bigger things. Those things in the winter, they kind of stop existing. And eventually in the winter, the plant relies on this local information, and those cells that are dormant, they do two things. They basically maintain their being alive, respiration, and then they can use the stored resources, either in form of the soluble carbohydrates, here soluble carbohydrates or starch, in their response to environment. Now, what is interesting about it is that all this, like all those 2 or 3 particular all those two or three particular activities related to sugars, they are under temperature control. So they are really under the thermal control. Temperature is a very important thing. Okay, temperature. So temperature is very important things. And in this case, they go to this, but also, as I said before, we believe, and it's a kind of a true, you can notice that, they kind of try to maintain this status quo. They don't want to limit, like drain out sugars or have too much sugars in the winter, so, or anytime. So what happens at the moments when they reach this kind of a this little moment when there is not enough sugar in the cell, let's say, at that moment the starch in the winter, starch has to be degraded and produce more sugar. or more carbohydrates. If they reach the top part, that's another zone when there is a moment when they have to say, wow, we have too much sugar. What do we have to do with it? Export it? We cannot, because we are dormant, no one is reactive. So we have to transform it back to starch. And starch. And what is it possible that they do it? So dormancy doesn't mean that they dead or they completely asleep, unless it's in the far Alaska when there is negative 70 degrees or negative 50 degrees C, and the cells are truly frozen. In most situations, especially in Mediterranean climate or climate, even in Europe, there is a lot of moments when there is the trees meander around 0 degrees and cells are still not frozen. And in these cases, there is this continuous activity. But however, when there is approaching frost, let's say, then cell usually digest the starch to produce more osmotica to avoid freezing. And when there is higher temperature, they do the opposite thing. And as I said, all those reactions, they depend on temperature. And how do they depend on temperature? So what happens is that they have this activation energy. Oh, by the way, the reaction, some of those reactions, starch synthesis needs ATP. So it's a energy dependent reactions, and degradation is independent, just degrade without additional input from the energy. So what happens is that when you analyze the activation energy and the response to temperature of different activities in the plant, in fact, interestingly, the starch synthesis has much lower activation energy, meaning that the response to temperature is much more steeper than the degradation of starch. So having those two different responses, what happens is that there is, at any given time, if there is a certain amount of pathways that allow starch to be synthesized or starch to be degraded, at any given situation, the number of those or concentration of those pathways, they can find an equilibrium. Basically, when there is a formation of sugar or carbohydrates, soluble carbohydrates and starch, they're in equilibrium, meaning that they don't change, okay? So there is this, what we would call kind of a temperature, equilibrium temperature, when there is no change in the amount of sugars. But this is like super specific one temperature only, yes? Because that's how cell works. So what happens if temperature changes? So if temperature changes, suddenly the same number, we shift down this pathway, okay? We shift down, let's say green here, or we shift down this pathway, and suddenly we are in the, to a new temperature, we are in the disequilibrium, meaning that at this particular point, or better do it here, we are in the disequilibrium. It means that in this particular point, the starch degradation, this one here, starch degradation will be higher, we have higher rate than starch synthesis, which means that there will be accumulation of SC. So SC, soluble carbohydrates, will start to accumulate in this point. If we go above this particular temperature, there will be SC, concentration of SC in the cell, we start dropping, meaning that there will be less. So we will go to this behavior here caused by the temperature. Now, if this happens, meaning that, if this happens, then plant has to respond to it because it doesn't want to maintain, if those prolonged temperatures exist, it means that there is a continuous shift to this lower level, to this level here, that there is a shift to this level. Eventually, if plant would like to maintain the average concentration, this point to maintain the status quo of soluble carbohydrates, it will have to change the number or amount of those enzymatic pathways. So it will have to shift, let's say, decrease the amount of the degradation path and increase the synthesis path to meet this requirement here. You see here, let's say, or this point here, they increase the number, concentrate, so produce more starch synthesis pathways, and then reduce the number of the starch degradation. So allow degradation of enzymatic pathways for synthesis, for degradation of starch, and synthesize pathways to increase the amount of starch producing pathways. So this is an interesting thing because and in fact, when you go to all the literatures there is some evidence of it when people analyze the activity of enzymes during winter in apples, they found exactly these patterns that there is a big shift in number or activity of two different pathways during the winter, through entire winter. So what it does ability, this produce ability for plants to, or not ability, but what we think about it that if there is a heat in the air, like there is so in diurnal basis, we have to remember the days, the diurnal temperature is like this, yes, continuously. So there is a temperature and there is a time, and diurnal temperature looks like this. So basically this, let's say that this was the equilibrium time for the plant to have this enzymatic reaction meet at each other at one particular point. There is really never true equilibrium, because anytime it's above this time, we have more accumulation of starch, and whenever we are below this line, we have more accumulation of sugars in the cells, soluble sugars. In the winter time, like let's say when we are approaching winter, you can see that this equilibration temperature that is in the spring, or sorry, it's in the fall, is at this moment. However, the temperature drops down, so it has to be chased. This equilibration has to be chased continuously by adaptation of those enzymatic pathways. And this is the average temperature of the, let's say, this is how the average temperature of winter in California look like. Average temperature with diurnal changes. Yes, so this, like you can see that these are the days, exact days in Davis, California. So you can see that there is this line. It's an average temperature. So this equilibrium has to be chased. And why it has to be chased? That's the idea that whenever there is a heat, it accumulates. There is a degradation there is a formation of starch, so the SC decreases. So the response of the organism, or the response of cell would be to accumulate more pathways that will increase SC, meaning they will degrade the starch. And opposite is here, when you have the chill response, is that it accumulates SC, so there is more soluble carbohydrates in the organism. And the response would be of plant to maintain the equilibrium, it would be to produce more starch-forming enzymes. And if those processes are slow enough, in the summer, you can envision that these are very fast processes, that this entire genetic information that produce or responds to the environment is very, very fast. Maybe it can adjust even within a day or within hours. However, in the winter, when the cell is dormant, those processes are super, super slow. So it means that there is a lag time in accumulation of and degradation of those pathways, eventually making possibility that a number and amount of the enzymatic pathways can be the true clock of the mechanism that is responsible for activity, or for counting up the time in the winter. And when we look at whether it's true that plants maintain the soluble sugars, we analyze hundreds of trees over a long time, over several years in California during winter, for 3 different species. And this is for almond, in fact. No, this is for pistachio. And you can see that in the winter, soluble sugars stay relatively flat, a little bit higher, and there is a drop in starch content in the plant, meaning that there is a loss to respiration that continuously is being regained from starch during the winter in California. So there is some sort of an activity in those dormant cells. This is in branches, not in buds. Now, so to better understand it, we decided to use this kind of approach here, this kind of a synthesis of model, like a very simple approach of model, and generate a model of not predictive model, but basically look at how the sugars in plant will behave and what can lead to bloom. And the model was we published this with our few years ago, and basically it's very similar to the system when we have the starch, then that it's being degraded to sugars, and then sugars being degraded to maybe form starch. So this is like this activity that goes in the plant, and there are the enzymatic reactions here, concentration of 2 enzymatic groups. enzymatic degradation and degradation and synthesis here, and degradation. And when they act over time, because of the temperature, the accumulation or degradation of amount of those pathways is kind of a decisive element of what happens in the plant on the long term. And when we create a model which is very similar to the dynamic model, except that we don't have accumulation and accounting for specific times. It's just time continuum, when we basically analyze the presence and absence of those enzymatic pathways and their activity on the sugars. Eventually we can predict the behavior of sugars concentration, projected sugar. This is projected soluble sugar concentration in the plants for different years with different temperatures, starting at the bloom time. So we start at the bloom time with some sort of an average concentration in the cell. And then over time, in the fall, there was some warm days. So we have formation of starch, and then plant responds to it, trying to force accumulation of sugars. But then it eventually reaches very high levels because it cannot continue to produce enough starch synthesis pathways in that zone. So there is like a lag time with production of starch synthesis pathways. So there is an accumulation of sugar. And then at the, in the mid of the winter, there is the temperature changes in the way that it's, from cooling starts to warming up. And so there is a sudden decrease in the starch concentration. So there is soluble sugar concentration. There is a decrease over time due to the fact that there is a warming temperature and there is a lot of starch formation. So there is a lot of starch synthesis that responds to temperature very high, which force suddenly this drop in the sugar concentration. And at one point, what we assume in our thoughts is that there is a level of sugar that forces bloom, because if there is no bloom, plants will die. Meaning we didn't care about vegetative or flower part at that point. We just said there has to be a bloom time. The buds have to break, because if they don't break, they will die out of starvation. So there is something what we call starvation level. And this starvation level can happen at different times, as you can see, depending on the winter projections. Like cold winters, we have a different effect on this starvation moment versus cold winters. And the way that those interplay between hot and cold temperature is very much, every winter has something different. So saying, 2 hours of heat will do this, or 3 hours of heat do this, 2 hours in the morning acts differently than afternoon. Yes, but in this case, it's all taken care together and produces this kind of a shape of response of plant in terms of mechanistic approach to, or not mechanistic, in terms of projection, what happens in the energy source in the cell. Now, if we run this model for multiple years and decided, okay, let's just take the real bloom time and run the model over long time, I think I will be like way over time. Then we can predict.
I don't think it's an option. So mind you.
Well, what do you mean? Zoom is never ending. You can talk and
yes,
talk.
But apparently they have some time limits.
Okay.
So
then, all right. So I will start talking much faster then. So what you can do, you can parameterize this model and see whether we can predict almond, apple, pistachio, or walnut bloom over many, many years. And when you parameterize the model, you kind of get an interesting process that yes, we can, meaning that there is a pretty good fit for many, many years over times, when we just run this model analyzing sugars and enzymatic reactions in the cells, and we can, not analyzing it, producing the parameters for it, and then see whether we can, to a certain degree, predict the real bloom time. Obviously, this, as we heard from speakers before, there is a lot of error, because all those data that we have on bloom time, it's somebody's observation saying, I think it's 50% right now. I think it's 10% of flowers are blooming. So there is a lot of error related to the fact that we don't really know how people estimated this bloom time. But still, the fit is relatively good using just analysis of sugar content and parameterization of the changes between starch and sugar in the plant. And this was done for almond, apple, pistachio, and walnut for California. This is California, 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 and Washington State. And in the apple, we have almost 100 years of data of bloom time. And when we look a little bit closer, we can say that in typically cold winters, how our model behaves in typically cold winters, we can predict that the hot and cold winters mean there's more hot, like average temperature of winter was warmer than particular year, or average winter was colder than the average of all the winters that we used. And for this particular things, you can see that projection and actual observation bloom dates, they pretty much stay correct for both winter, for cold and the warm winters. Now, there is the, where did it, so, but it's also worth to notice that to produce this particular points, we used like a starting point from here. You can see the starting points of carbohydrates was very, we just assume average carbohydrate from this state average. So what we have this carbohydrate observatory when we collect data and analyze carbohydrates across entire state from hundreds of orchards and thousands of orchards, not half a thousand, 500 orchards. And you can see that we just took this average temperature for each winter to start our model. However, as you can see, there is a big range in the winter or in the fall between, like high concentration of sugars to start the winter or low concentration of sugars to start the winters. So there is a difference in it in the state, across the state. So the next question for us was to ask how this initial concentration of sugars affect the bloom time. And interestingly, what you can do, so this is the average concentration for different years. Like this is the concentration, for 2000. So this is for 2017. This one is for 2018, and this one is for 2019 for almond, yes? And then when we start taking those concentration and plug them into the model, we realize that in certain areas we have the model predicts, like for example, increased concentration of sugars, soluble sugars, shifts the date to get young, to bloom, like forces plants to bloom earlier in the model, of course, not in real life. But if you put this in a very cold year or typically cold year, it would the same values, the same values of carbohydrates, we start to shift bloom time much later. And it was true for pistachio. And although in pistachio, you can see that increasing amount of sugars will force bloom earlier, and the same for walnut. Although if there was a lot of sugars in the very cold winter, eventually it will start pushing it backwards. Which is an interesting thing, because it means that sugar concentration at the initial part of the year, that typically it's a pattern of temperature in particular year, can have a different effect on the bloom time, which is kind of mind-boggling, because plants prepare those resources from the summer, and then they start the winter, and this is why we call it winding up the clock for bloom, because they prepare those carbohydrates and then they shift it and start counting hours. But depending on what amount of sugars they will count an hour differently. So there is no just simply saying this whether we do this whether we do this. It's depending how tree was prepared for the winter. And when we just play with numbers and say, okay, let's just feed the particular sugar concentration to the model and see whether we can match perfectly the dates from 100 years. We can, like for apples, we can match perfectly. Like dates can be perfect. It's not prediction. It's not predictive model. It's just that by playing with the amount, how tree was prepared for winter, we can get a prediction. We can get, not prediction, but we can estimate the time of bloom from this, when we just assume one particular concentration, to this, where you can say we can basically find a preparation of tree that will perfectly show on the prediction model predicted bloom time with prediction of with real bloom time in the field. And so now when we plug it in, we say, okay, so what are these sugar concentrations that we had to use to make this perfect feed? And when we did that, it turned out to be that the sugar concentrations to make the perfect feeds are really within the same zone as true sugar concentration that we observe in the field. Okay, those are the same concentration, walnut 40 to 120, and walnut in our model was 50 to 120, exactly the same concentration. So it means that indeed, the fact that they have this variable amount of sugars to start with, we can probably predict that those sugars can be part of the process that decides, we together with the stochastic aspect of weather, can decide when tree will bloom in this particular year. And even if you have the same exact weather conditions, the year to year for winter, you will know, I mean, you will not notice, but you will notice, like, there might be a different outcome depending on where they start. If they started here, okay, this particular winter, if they have less at the sugars at the beginning, despite that winter would be exactly the same, they will bloom at different time the following year when the same winter conditions, but different starting point, like say. So this would be a shift in it. And interestingly, what we did, we ran the analysis for all the years that we have of the bloom time, and asked the question, how does the change in the carbohydrate will impact the bloom in particular year? So this is like each column here is a year, and it has particular aspect of temperature, which is associated with the year. But we just said, this is the true bloom time. Like this one is the true bloom time here. This is a true bloom time here. But what if we change the starting point with carbohydrates? If we ask the quick tree to have less carbohydrates, what would happen? And in fact, in the apple, it will delay the bloom. So the less carbohydrates they have, in most cases, they delay the bloom in model for particular winters. For example, for almond, it's kind of opposite. You can see that almond will, in most cases, when we increase the concentration of sugars in the tree to starting point, it will result in delaying. But so the lower concentration will go and result in earlier bloom, but not always, because in some years it will do the opposite. So it's really a pattern related to particular year temperature and starting point. But on general, all those trees start their concentration somewhere in the middle, like here, and bloom in the right time. But if they start too little or too much sugars at the winter, then it will shift. Now, this is further shown here when we show that the early, normal, and late bloom will result in depending on the sugar concentrations. In almond, late bloom will be enforced on average by higher concentration in the fall, and in apple, the late bloom will be enforced by the lower concentration in the apple in the particular place. In walnut, it's also lower content. In pistachio, lower concentration will enforce earlier bloom, and later bloom when the high concentration enforce the early bloom, which suggests that maybe there is a chance to prepare trees for the changing climate by understanding how they respond to the bloom, and then prepare plants with particular levels of carbohydrates. Because if we want to, for example, force plants to go early in colder winters, we should, for example, for apples, prepare them with high concentration of sugars, soluble sugars. And if we would like to do the same for the almond, we might have to do the opposite, prepare them with with low concentration of sugars to enforce the earlier bloom or the bloom that responds to the climatic changes. Okay, and I think that's, I'm like, how much time do I have?
You don't.
No, we're done.
Are we done? Okay, so now there are questions.
Now it's my time to speak, no?
Oh yeah, it's your time to speak. Okay.
Okay.
But I have time for questions, no?
Yes. I just like to point one thing, and then there's questions. So we first called the model, later we called it the CT model, the carbohydrate temperature model, but first we called it the dynamic model. because the main components, these two curves that Maciej demonstrated, one for starch synthase and one for starch degradation, they are actually the exact curves that Amnon Erez published in the original paper. So, but the only thing we changed is that, so there's this response to temperature, but then there is a constant, so it's alpha to begin with. And we figured that that is the amount of enzymes. So we're now changing this alpha. So instead of using a fixed dynamic model, we're using a dynamic model. And that enabled us to not do this later component in Amnon's model where he counts a stable form. So there's, whenever something happens, it ticks, and then that's a chill unit. In our case, sort of you can think that the chill unit is this ends the pathways increasing or decreasing in the amount of enzymes that they have. So it's very much
it's a continuum. Like I would say that we don't have any, because you can also continuously degrade the enzymes due to the fact that we believe, I mean, that's biology. You can't just accumulate something and it stays there, because there is always a degradation. So there are those enzymes that basically they being accumulated pathways, but at the same time there is a process of degradation, and they can change their composition at the same, basically just the proportion, how many of each one is there, not necessarily how many of them there is. So it's not accumulation of something, it's basically changing proportion of enzymatic pathway concentration that degrade or accumulate starch.
And also Amnon, in the original paper, he has there in parentheses a comment that this, whatever the process is, it might be related to enzymes. And so like this really guided the way we first developed the model. Another thing just to think about, because Eran was asking me here, so why would almonds bloom later and then apples would bloom early? So just think, so almonds are early, they're very early bloomers. They can only benefit from sort of postponing the bloom time if they could. Better weather, more pollinators, and so on. And apples are actually very late bloomers. They can benefit from a shorter dormancy if the trees could sustain the metabolism in a longer period where they're now in bloom, but they might not be photosynthesizing. So that could be a lot about these evolutionary strategies to cope with the natural habitats of these species. But that's for another work. But not all trees. Anyway, now questions.
I have a question.
Yeah.
I mean, go ahead. Like there is no like, oh, I have a question. It's not classroom.
What about the mobilization of the sugar from the storage part of the tree?
I mean, this is what we're talking about. This is a storage part. As I said, I don't personally, I see the tree as a unit, not as a bunch of independent things. So if as I asked the question before, if there is a decision in the tree, we start blooming, there is not a lot of time to say, oh, this branch will bloom, but stem is dead. There will be no water, it will die. So everything has to wake up at the same time. So when you say mobilization from distal part, yes, there is a mobilization at the distal part after the bloom when they start to redistribute the things. But the local information to say, that's the end of winter. Now I am switching from dormancy to activeness. That's a time that tree has to make an entire decision. It's not just a decision of the bud. Bud can just say, oh, I'm growing, and it will die. So the decision has to be everywhere. So it's not just about redistributing and supplying. Yes, at the end, there is a supply. Like once buds start growing, it has to get sugars, and it has to be pathway already existing to do it. But decision when entire tree has to wake up comes from local information. It's like, it's a cellular automata. That's how I see the trees. It's those cells, they don't know what happens 2 meters apart. unless there is a transport. And in winter, this transport is very, very limited. So in the spring, they have to make a decision. I'm waking up, everybody around me start to wake up. And once we're walking up, like we finish the dormancy, we can start growing. So the aspect of bud break and the aspect of trees start putting new vessels and so on, it's an aspect of a final that's it's beyond the waking up call. The wake up call is a little bit earlier to everybody.
Okay. But then there is still the part that we didn't dare yet to include in the model, and that's the transport because of the thermal gradients that we published before. So we figured that actually when the canopy sees this heat, the roots are still, they're still very cold. They don't see these diurnal fluctuations. So the average temperature is the only one to affect them. And in this case, the roots are now, they're degrading the starch when the canopy is accumulating the starch.
Or is right. There is this at the end. There is this like potential for redistribution of sugars and carbohydrates in the tree, but at the beginning it is a little bit more of a. Once the tree has to wake, so it has to wake up.
But when we first wrote the model, we thought we'd include this part, and then it got overwhelmingly complicated. And then Jonathan, who asked the question, was a master's student who was supposed to develop the next stage of this model, but then he went to do other things. So Matcha, this is.
I in my master in all labs, I asked this question: if the sugar concentration in the fall will determine the bloom, and in my project I didn't get a yes answer to this question.
Sorry, I did not understand. You were like breaking off.
Well, Jonathan we actually did these experiments and they did not work in apples. This is why I don't work with apples anymore until you came up with a data set in Washington State. So, so Jonathan, maybe, yeah.
Okay. Can I ask a question?
Go ahead.
When you speak about carbohydrates, there must be an effect of the former yield and the amount of crop that the tree was bearing, especially if you speak about species that produce their fruit very late. Like certain varieties of apple, for instance, if they have very heavy yield, the tree won't be able to recover. And what will be then the effect on the dormancy of the buds of this tree? When in two cases, one, it bears very heavy yield compared to light yield. Do you see any effect?
So I don't study apples. Yes, there is a negative correlation between end of summer amount of starch and sugars in the trees, like almond and pistachio and crop yield. However, interestingly, this thing disappears completely by the time when they go to the fall. There is really no effect in the fall, whether there was a heavy crop or not. And we tested it on 500 orchards, and there is no correlation. So the amount of sugars and starches in the plants in the pistachio, almond, and the walnut is in October is completely independent of the crop. It is there is a dependence between in August, but not in the October. So we couldn't find this kind of an information. However, still the amount of sugars were completely different in those trees. So it means that there is aspect of management or weather or particular local conditions that affect the amount of carbohydrates that are being stored in the cells for winter, but it is independent of the crop.
Okay.
Maciej, if I got it right, you said that blooming is in an NSC threshold in almond, let's say. So if I will manipulate the carbohydrate status during winter or at fall, it's not supposed to have a positive effect on yield because it always get to the threshold?
So the threshold, it's not NSC threshold, it is soluble sugar threshold that causes the bloom. And, but it depends what was, yes, it depends what was before in the plant. So there is an interesting, we run right now models using the carbohydrate content and the predicted yield. And there is, for several species, almond especially, and in pistachio, there is a negative correlation between, there is a positive correlation between amount of sugars in the winter, especially in the January, in the branches, and the predicted yield. So the higher, and yield potential, the higher amount of carbohydrates in the winter, the higher the yield. But it's independent of the bloom time in this case. It's basically saying, okay, there is a more carbohydrates in the winter, there is higher predicted bloom, but, sorry, higher predicted yield. But it doesn't mean that there will be there's so many other components related to weather and management that affects it, but there is a correlation with it, but it's a kind of independent from the predicting the bloom time.
Okay, so carbohydrates at bloom time are a limiting factor?
For the yield?
Yeah, for yield.
So, I mean, it's not a limiting factor. They affect the final yield, yes, but it's not a limiting factor. It's a necessary. It's a component that of the final productivity.
But also,
but there are other components that are the limiting factors. Like if no matter how many carbohydrates you have, if there was no pollinators, there will be no bloom. Yes, so there are other things that can affect it. So carbohydrates are necessary, but not sufficient to the high bloom. Okay, so
high yield.
To high yield.
You think of aspects of, for instance, cross-pollination for almonds, right? So you want them to bloom together.
Let's say in a self-compatibility cultivar.
Well, you're the only one to have the SF. No, I'm joking. But anyway, I'm just saying that the time of bloom, if you affect the time of bloom, you affect yields eventually, right? Because you affect cross-pollination, you affect the weather, or like the time that trees bloom means that they might see different weather conditions and bee activity. So, and then as Maciej pointed out, so it's only a sort of a short metabolic deficiency. Later, there is the amount of starch there is the energy that is the residual energy for the rest of the processes before you have photosynthesis. So let's say what plants, the optimal conditions are when they hit this metabolic deficiency while they actually have a lot of starch to go with later. And the trees which are depleted, say high yields or poor management in fall, would still be affected, independent of the fact that you always respond to the same soluble sugar concentrations.
But obviously we have to admit that it's whatever we say about this carbohydrates and so on, it's not that it's a final model of everything. Like we know that there is a genetic component, there are hormones, there are other stuff, but the moment when cell wakes up, that's where all the expression activity suddenly goes from very close to zero to very high element, and that's where all the other aspect of the plant function starts to be there, and all the processes speed up and go to a different level. But during the winter, there is this very, the respiration of branches in the winter is 1/10. No, we measure it's not 1/10. It's 1/20th of what is in the respiration during the spring. So there is a big change in how plant utilizes the energy and how it goes toward the how it changes during the bloom time.
Okay, thank you.
Okay, I think we should end the session.
People are disappearing
because people are dropping out of Zoom.
Okay.
Thank you for participating. This session was important for me, considering the speakers. I actually asked the people I really wanted to listen to
except me.
talk. It was a great trick. I get, you know how to contact every one of us, of the people who spoke. So if you think the sessions, the talks were intriguing enough. Anybody else wants to conclude?
I don't know whether conclusion, just saying thank you for invitation, or and I hope that like 2 or 3 years from now we can meet in person.
You know, when we started, when they told me that I should organize a session, what they promised was that I can bring you all to Israel, right? So I was just thinking, oh, there you go. Machi is on the plane and Ike is on the plane, and then it didn't happen. So you missed.
That was the only reason we agreed to give these talks, right, guys?
Never agreed. You are the only one to not agree when you had to travel to Israel because you were too busy, and then agree when it was a Zoom session, which is a funny.
That's true. You're right.
Thank you for doing this. Yes.
All right. So if there are any other questions, maybe anybody has something? No.
Thank you.
Yeah.
Thank you.
Thanks, everyone.
Bye.
Thanks, Laura. Thanks for putting this together.
Bye-bye.
Bye.
Bye-bye. Bye-bye.